Today we will discuss the final figure of the paper or of the story where we have understood that in the in vitro study the authors or researchers of this paper has found out that IRF-1 modulations in the M1 macrophages will affect the translation of the RPS18 within the M1 macrophages, and simultaneously this RPS18 will then transfer or translocate to the PDAC pancreatic cancer cells where it interacts with the IL-3, and that IL-3 that where it interacts with the IL-3 and also upregulates the ILF3, not IL-3, ILF3, and those upregulation of the ILF3 will then trigger the increase in the lipid metabolism within the pancreatic cancer cells. At the same time also helps in the proliferations, invasions and migration of the pancreatic cancers that we have discussed through the in vitro studies. However, in order to validate whether their in vitro studies replicate in the in vivo model or not, they did their experiment in the they did their experiment in the mouse model where they use the orthopedic mouse model and uh, injected them with this M1 exosomes of different conditions. At the same time, they also injected these pancreatic cells. So we will start with this panel A, where they have used this control exosomes, that is exosomes from the macrophages, wild type macrophages, and overexpressions of this uh, overexpression of this control overexpression of this control. So in this case, or we can say overexpression of this negative control, where actually there is nothing is overexpressed, it's just a control sample. So in this case they can see that there is a, in the first case, that is they can see that uh, they consider the relative expression is 1, and also for the ILF3 they consider the relative expression is 1, as this is act as a control. And for the overexpressed IRF-7 exosomes, that is the M1 macrophage has this M1 macrophage is transfected with the lentivirus that has this IRF-7 plasmid that will lead to the overexpression of this IRF-7, and the exosomes collected from those macrophages are used in this second conditions. At the same time they use for the second conditions this overexpression negative control. So in this case nothing is overexpressed. So in this case, since the IRF-7 is overexpressed, they are expected that RPS18 will go down. At the same time, since RPS18 is go down, IRF3 will also go down. So this is what they see in the second conditions, that RPS18 is goes down compared to the negative control, and ILF3 is also goes down compared to the negative control. In the third case, they use where they knock down IRF7 exosomes. However, they overexpress RPS18. So once the RPS18 are overexpressed, the function of IRF7 will be negate or compensated by overexpression of RPS18. So once the RPS18 is overexpressed, it will then translocate to the pancreatic cancer cells and upregulates or promotes ILF3. So in this case, RPS18 is overexpressed through the lentivirus, and this increased overexpressed RPS18 will help in the upregulations of this ILF3. So this is the results that is in line with their in vitro studies. So, and these things they have also validated using the Western blot, where they can see that when they use the negative control, RPS18 and ILF3s are almost of same intensity. However, they have not provided the quantification of the Western blot. Once they use IRF7, once they knock down IRF7, they see that reduce in the number of once they overexpress IRF7, not knockdown. Once they overexpress IRF7, they see the reduce in the number of RPS18s. In IRF7 regulates RPS18, and once the RPS18 is reduced, they also see the reduction in this ILF3 compared to the wild type. And when they overexpress RPS18, the RPS18s are formed high because it has been overexpressed through the lentivirus, and the overexpression of RPS18 will cause overexpression of this IRF3. That is also shown in this figure. And they see the tumor growth in the mouse model at different time intervals, and in this case they can find out that in the third conditions where RPS18 is overexpressed, they see the tumor growths are all higher compared to the case where only IRF-7 is overexpressed in the M1 macrophages. And also in the negative control where nothing is overexpressed, the results are also looks like in similar to the third conditions. And thereafter they check the lipid metabolites or lipid number in the different conditions, and they have find out that lipid in the third conditions, lipid in the conditions where only IRF-7 is overexpressed is reduced, whereas lipids number when the IRF-7 is overexpressed, along with this RPS18 is also overexpressed. So they see that lipid numbers again increase and are in line with the lipid numbers in the negative control. And thereafter they check the tissue morphology. In this case also it is clearly evident that when RPS18 is also overexpressed along with this IRF-7, they see the increase in the tumor growth compared to the case where the RPS18 is not overexpressed. And in the panel F, they quantified the tumor volume. They quantified the tumor volume at different time intervals, and in this case they can see that when tumor volume are maximum in the case where tumor volumes are maximum in the case of this negative control. However, the difference between the negative control and the one with RPS18 is overexpressed is negligible, and the conditions in which this only the IRF-7 is overexpressed and the other overexpression is the negative control, they can see that there is a statistical difference, and then IRF-7 only the IRF-7 overexpressed macrophages exosomes shows a decreased tumor volume. And in order to check their proliferations and migrations properties, they use Ki67 staining, and in this case for the negative control they see that the blue populations are higher, and for the IRF-7 they see that these populations are lower. However, when they use overexpression of RPS18, they can see that Ki67 are expressed high, and from these images it is not clearly evident, so that's why they also quantified these images. And in this case we can see that when IRF-7 is overexpressed, only the IRF-7 is overexpressed, we see IRF-7 is overexpressed, along with that RPS18 is also overexpressed. We can see that this Ki67 positive cells again restores and are in line with the conditions where the wild type negative control exosomes and negative control overexpressions plasmids are lentivirus are used. So this indicates that their in vitro, whatever they find out in the in vitro studies, also seems to be applicable to this in vivo studies, and this could be the actual relationship that occurs in the human beings also. So I hope you understood their whole studies and what they have actually find out, what are the relationship between IRF-7 and pancreatic cancer cells functioning. If you need to understand more about these papers in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.